This is part 3 of the lectures on cell structure and function. And this slide shows you pictures of red blood cells, erythrocytes, when placed in isotonic, hypotonic, and hypertonic solutions, what happens to the red blood cells. And you can see what happens in the illustration, but you can also, if you look down here, you can see what the red blood cell actually looks like. This is a real image scanning electron micrograph. So it shows a lot of good detail. In an isotonic solution, which of course would be normal saline, we said that was 0.85% sodium chloride, that is not going to cause the cell to change size. Water will leave and water will enter, but the same amount that leaves, the amount that leaves equals the amount that enters. In a hypotonic solution, there's way more water outside the cell than in. So, like for example, if the cell were placed in a 0% salt solution, such as distilled water, that would be hypotonic, and the water would enter the cell and it would swell and it could burst. And that is called hemolysis when that happens. And then in a hypertonic solution, which would be a really salty solution, so say for example a 5% sodium chloride solution, the water is going to leave the cell and the cell is going to shrivel, and that is called crenation. Crenated is right here, but I'll write it crenation. Okay, checkpoint questions. I am only going to go over 8 and 10 because of time. So how would a decrease in the concentration of oxygen in the lungs affect the diffusion of oxygen into the blood? Well, if it was a lower concentration of oxygen in the lungs, then oxygen would diffuse from the blood back into the lungs. So less oxygen would diffuse into the blood. Here is like a air sac, a lung air sac, which usually has a high concentration of oxygen in it. And then this is an example of a blood cell, a blood vessel. And normally what happens is the oxygen diffuses into the blood, but you would have less diffusion of oxygen in the blood, or a lower rate of diffusion if there was a decrease in the concentration of oxygen in the lungs. And if the oxygen concentration in the lungs decreases enough, you can even have oxygen diffusing from the blood back into the lungs. 10, relative to a surrounding hypertonic solution, the cytosol of a red blood cell would be hypotonic. The fluid inside the red blood cell would be hypotonic in comparison to a hypertonic solution. All right, carrier-mediated transport uses a carrier protein in the plasma membrane to move ions or other substances, organic substances, across the plasma membrane. If the substances are moving from high to low concentration and no energy is required, no ATP is required, then the carrier-mediated transport is typically called facilitated transport or facilitated. All right, let me think about my spelling. Facilitated diffusion or facilitated transport. So that's when it is a carrier protein is used, but no energy is required. That's an example of, is that how you spell facilitated? That's an example of passive transport. But carrier transport can also be active whenever you're moving a substance from low concentration to high against its concentration gradient, and ATP is needed, then that would be active transport. So carrier-mediated transport can be passive or active. Carrier proteins are specific to their substrate. For example, if they carry glucose, they will not carry other simple sugars or other chemicals. And many transport only one ion or molecule at a time. Some can do two or maybe even three, but that's about it. Some carrier proteins carry two solutes at the same time, and when they move them in the same direction, it is called co-transport. When they move the two substances in opposite directions, it is called counter-transport. Examples of carrier-mediated transport are facilitated diffusion, and this would be no energy required, and it would be a passive process, and then active transport, which does require ATP. Both of them require a carrier protein to move the substance across the plasma membrane. Facilitated diffusion uses carrier proteins to passively move larger compounds down their concentration gradient, meaning from high concentration to low concentration. The carrier protein binds to a receptor site on the compound binds to a receptor site on the carrier protein. It changes the shape of the protein, and that allows the compound to move across the plasma membrane. Glucose and amino acids are examples of nutrients that are transported this way, and facilitated diffusion is limited by the number of carriers. So even if you still have a higher concentration of glucose outside the cell than inside the cell, if you've already used all of the glucose carriers, you can't bring in any more glucose. So the number of carriers does limit how much of that substance can come into the cell. And this happens, we notice this whenever a diabetic has glucose in their urine. It's because there was too much glucose to be taken into the liver cells or the muscle cells, and so the glucose remained in the blood, and then it would be that excess glucose would be excreted in the urine because there were not enough carrier molecules, carrier proteins, to remove all the glucose from the blood. So this is an example of facilitated diffusion. You can see the concentration of glucose is high in the extracellular fluid and the blood, and it's low inside the cell. So the glucose will bind to the carrier protein, and then the carrier protein will change shape and bring the glucose into the cell. Active transport is going to be exactly the same process, except energy is required because the substances are moving against their concentration gradient. They're going to move from where their concentration is low to where their concentration is high. That means against their concentration gradient. There are ion pumps that transport ions, which are charged atoms. Sodium, potassium, calcium, magnesium, and chloride ions are shown here. And there are exchange pumps. The sodium-potassium exchange pump is extremely important, and this is going to show you how it works. We're going to need to understand the sodium-potassium exchange pump in order to understand nerve impulses and muscle contractions. The sodium-potassium exchange pump helps to maintain a certain voltage or a certain potential across the plasma membrane. So what it does is it pumps 3 sodium ions out for every 2 potassium ions in. So it's pumping more positive ions out, even though there's already a high concentration of sodium outside the cell. So sodium is moving against its concentration gradient. And there's already a high concentration of potassium in the cell, but the pump continues to pump potassium into the cell toward its higher concentration. So this is an example of active transport. And what that does is, since there's more positive charges outside the cell and less inside, it makes the inside of the plasma membrane negatively charged relative to the outside. The outside is positively charged relative to the inside. That's what I mean by there being a voltage across the plasma membrane. And that is very important to maintain that particular voltage, because nerve impulses and other processes, muscular contractions, will not happen unless that voltage is maintained. Another type of active transport it's not active transport, but it does require energy, is vesicular transport of substances through a vesicle, a membrane enclosed sac called a vesicle. And there's endocytosis, transporting the substances into the cell, and exocytosis, moving the substances out of the cell. Endocytosis brings materials into the cell through a vesicle. The vesicle is actually pinched off of the plasma membrane. It forms from the plasma membrane. There are three types of endocytosis: receptor-mediated endocytosis, pinocytosis, which is like cell drinking, and phagocytosis, which is like cell eating. Receptor-mediated endocytosis brings in a specific type of molecule that binds. The molecule is called a ligand, and that ligand will bind to receptors on the plasma membrane surface, and then the plasma membrane will form a pouch, an in-pouching of the plasma membrane, and it forms a coated vesicle coated with the ligands and the receptors. And then the ligands are removed from the vesicle and the receptors are recycled to be used again. The ligands come into the cell. And two substances that are transported by receptor-mediated endocytosis are cholesterol and iron. And this shows you how the process works. You see the receptors on the plasma membrane surface. The ligands are these little round purple molecules. They're binding to the receptors here, and you can see the in-pouching or in-folding of the plasma membrane, and it finally pinches off, and now you have the coated vesicle. The coated vesicle in this case is fusing with a lysosome, and a lysosome, remember, contains digestive enzymes, and that lysosome is removing the ligands or the chemicals from the coated vesicle. The vesicle is going to come back up and fuse again back with the plasma membrane, and that allows for the plasma membrane to stay the same size. A vesicle pinched off, but the vesicle then fuses back in with the plasma membrane, so whatever was lost is regained. Pinocytosis is cell drinking, and it just means that the material brought in by endocytosis is fluid. And phagocytosis is cell eating, and that just means the material brought in is more of a solid. Sometimes a cell, like a white blood cell, will contain cytoplasmic extensions called pseudopodia. They will surround the object, and then these would be the pseudopodia. For example, this is a white blood cell, and this is a bacterium that is entering the white blood cell by phagocytosis. Once it is inside of a vesicle that pinched off from the plasma membrane, a lysosome fuses with it. That lysosome breaks down or digests the bacteria, of course, killing it. And then exocytosis causes the membrane to fuse, the vesicle to fuse back in with the plasma membrane and to release the waste products of the digestion of the bacteria. Exocytosis is the reverse of endocytosis. It just means that a vesicle fuses with the plasma membrane and excretes a product. This is a table that is a summary of passive and active membrane transport processes. and it summarizes diffusion, which is passive, and factors that affect the rate of movement, the steepness of the